All right, hello and welcome everyone to our first of 4 sessions where we will learn from experts on applying complexity methodologies in research. You can learn about all of our sessions through the link in the chat. Here we go. My name is Joey Eisman, and I am a doctoral student studying at Temple University and the graduate student representative of the Complexity Theories in Education SIG. The purpose of the SIG is to apply, advance, and extend complexity theories to inquiry, research, theory, and practice in a variety of educational contexts. I am pleased to be joined this evening by Dr. Daniel Russell. Dr. Russell is an associate professor and serves as the graduate program director for the PhD in kinesiology and rehabilitation at Old Dominion University. He earned an undergraduate degree in sport science from Manchester Metropolitan University in the United Kingdom and continued his studies with MS and PhD degrees in kinesiology from Louisiana State University and Penn State University, respectively. His research is guided by complex dynamic systems perspectives, leading him to develop expertise in measuring and analyzing different aspects of human behavior over time, from human movement during walking to eye sequence gaze while studying. During this talk, Dr. Russell will introduce concepts and methods for analyzing time series of interval ratio and nominal level data. A majority of our session will include Dr. Russell's presentation. If you have any questions, please post them to the Jamboard linked in the chat. Now.
Thank you, Joey, for that.
Yes, please. So without further ado, Dr. Russell.
Thank you. Okay, hopefully you can see my slides. I'm going to talk today about variability in time series data, and we're going to go over concepts and methods. So, as Joey said, I've spent a while sort of learning concepts and methods, looking at different time series data, and particularly sort of examining variability in those time series data. So I'm going to begin with the sort of the general concept of variability is assumed to be noise, and I'll highlight to you why I don't think that's the case. We will do some time series analysis that reveals structure in variability, and this can be used to distinguish groups. So that will be sort of focusing on interval ratio data, and that's sort of more from my background of sort of human movement data, because we can collect lots and lots of data which makes it amenable to some of these analyses, but I'll sort of describe how they can be used in other ways. And then I'll finish off the talk looking at symbolic dynamic analysis that reveals structure in nominal sequences, and this is in particular we looked at gaze sequences during studying behavior. So I think one of the sort of main things that sort of a complex dynamic systems approach sort of does is to flip this sort of notion of variability on its head. So, rather than considering variability as noise that's sort of on top of a deterministic system, sort of where the sort of structure's there and there's noise sort of hiding it, a complex dynamic system considers how structure emerges from a system of many parts, so that this noise variation is really embedded in the data that we see. So from my research, we look at human movement, and an example is walking. It's obviously a very important skill for us to perform, and we can seek a model of walking, and it's sort of not a far leap when you study a little bit of physics to think that a pendulum model might be a good explanation of walking. And if you've studied a little physics, then you'll know that pendulum, a simple pendulum model allows us to predict the resonant frequency or resonant period, so the time of each oscillation of that pendulum. And of course, this has been very useful for us as it's allowed us to create clocks that sort of time out our seconds of the day. So we can say, is this, can we model human walking as a pendulum and predict the stride interval, stride period? Okay. So when we do this, we, with the science, we take repeated measurements of any sort of phenomena. We're looking for repeatability. And you can see here is some data we've collected from a participant where we've got knee angle data. And the knee angle, you could see, sort of varies across these different strides. It's not exactly the same angle each time it sort of turns. And we can measure the interval of time in between those peaks, and that tells us the stride time. And if we do that, we get data that looks like this. So we can see these stride times are varying, and this is for an individual walking on a treadmill for 30 minutes. You don't need to collect quite that much data, but there's a sort of thing we can do in our lab. We can have people walking for a while, and you can see that there's variation. And these are 3 different speeds we asked them to walk at, just based on their preferred walking speed, 90% and 80%. I think it's quite clear there's variation on this in their gait times. This is on a motorized treadmill, which is constraining the speed, and there's no other environmental manipulations here. So nothing else is changing, but there's variation in the person's output. And this is not just simply measurement noise. We're able to measure this pretty precisely, and we're seeing variation that is well within our ability to measure it. And let's take another system. So this is an individual's heart rate, their beats per minute, collected over a period of 15 minutes. And this is a resting individual, and they are healthy. And I think it's, again, quite clear that there's quite a large variation. I have a smartwatch, which tells me my heart rate, and I can see how it changes. It obviously changes with different environmental things, or whether I've had coffee or doing different activities. But if you rest, it changes too. There is inherent variability. And of course, this is present throughout biology, and is in a critical feature of natural selection. There would be no evolution without biological variation. So when we behave, when we move, it is inherently variable. So the standard approach to variability is to assume that it's in the form of white Gaussian noise, that it follows a normal distribution around some sort of mean. If you think about in terms of measurement theory, we are expecting, if we measure something repeatedly, then we are able to see or approximate the true score as the mean, and then we get sampling error around that mean that's just thought to be sort of variation and just sort of noise. I think a key thing to sort of understand when we look at this is that we have disrupted the sequence of values. So each stride time, when you do a distribution like this, you are ignoring the sequencing of those stride times, or any measure that you get. So anytime you do a frequency distribution, you're ignoring the sequence in the data, and you are and then you're collecting a sort of an average or a magnitude of that sort of variation around the mean. So here's some data that we saw the top graph before of a healthy individual and their heart rate. And the bottom graph is an individual with congestive heart failure. I think it's quite easy to see that there is some sequence in the heartbeats there. But if you just look at the mean and the variance, you see that there's no difference in those. So we're not able to distinguish any structure, any difference between these two time series using linear measures. So we need to sort of think about this. There's some structure there. How can we detect it? So there's a few different ways of doing that. The first thing I think we don't do enough of throughout the whole data collection and analysis is to look at the frequency distribution. I know in statistics we teach people to sort of assess this, but we rarely actually do a histogram and look at the frequency distribution. And as you can see here, we have 3 signals. So the top left is a straight sine wave that's just a created signal, a created time series. The middle one is a human pressing on a cell, load cell, with their index finger, trying to produce a constant force. As you can see, we are not very good at producing a constant force. There's a lot of variation in there. And then the top right is a created random white Gaussian noise time series. So only the middle one is actual real data. If we do a frequency analysis, do frequency distributions there, we can see that we can tease out the sine wave is different than the other 2, but the other 2 sort of approximate normal distributions. So that's sort of the first thing we can look at. But as I identified before, we're sort of removing any sequencing, any structure in those sequences. So the question then is, are there ways that we can look at that structure and sequences? One approach is to look at a recurrence plot. So in a recurrence plot, you take a signal, any time series xt, and you plot it against a lagged version of itself, xt plus 1 in this case. So you can see that for the sine wave, it's this perfect correlation, perfect relationship. You can predict the next value in the time series based on the previous one. If you take the right-hand side, the white Gaussian noise, you see that it's a very sort of a dispersed sort of cloud of data. So we don't know from any data point where we're going to be next. There's really little predictability in that system. But in the human isometric force, we saw that sort of random variability in that middle top trace. And actually, there's a lot of structure in there. It actually produces a correlation in that recurrence plot of 0.92. So there is certainly a strong relationship between current force and subsequent force. So recurrent plots start to tell us something about the structure. And this is something that you don't need lots of data for. You could do with very small data sets. A technique that a lot of people may be familiar with is autocorrelation. So that's really what we sort of were able to see in that last plot, was a sort of a relationship between a value and its subsequent value. And that can be calculated as an autocorrelation. So a correlation, you correlate a signal, a time series, against a time-lagged version of that same signal. And you can do that with different lags. So you get the plots that you see on the bottom there with lags of 0.1 second, because this data was collected at 100 hertz, 100 times a second. And so you can see correlations going from, for the sine wave, going from 1, perfect correlation, down to 0, and then down to a -1. For the human isometric force, you can see that there is correlation that decreases over time to that 0.20 lag. It drops down to about 0. And you can see in white Gaussian noise, there is no correlation. So that each value is independent of any subsequent value. So that's a nice sort of linear approach that you might be familiar with from correlation. So when we look at time series and we see that they're noisy, there's 2 possible things that could be going on, or of course some combination thereof. It could be a random process or it could be a deterministic process that's underlying this. So you see a sine wave on the top graph, giving you a phase plane plot is simply a plot against its first derivative, so a value against its first derivative, its slope, and you can see that it's a very simple, what we would say, attractor, a limit cycle attractor. In the middle graphs, we have Lorentz equations. So these are deterministic equations, 3 equations, and you can see that the graph looks a bit strange on the left-hand side. And when you plot that out, you can actually see that it produces this sort of nice sort of pattern effect, and that's an example of a chaotic attractor. On the bottom graph, you've got white Gaussian noise. You see how sort of random it looks. And when you do a plot against the first derivative, you see it truly is random, right? There's very little relationship between a value and its subsequent value or the rate of change of those. So we've seen that sort of noisy data can be produced by sort of these random signal or a deterministic signal. And these recursive plots, again, here in the bottom, you see help us to sort of tease apart those particular signals. You can see on the random signal how any value at xn, it can change to anywhere else in this space for xn plus 1. So there's no real relationships there. But for deterministic chaos with a logistic equation such as this, we see a nice clear regression, a nice clear structure in our recurrence plot. So when we're collecting sort of, so we've got these 2 dynamics here, this sort of, there's 2 ranges, random noise, deterministic chaos. Now, the mathematicians have it easy because they can create all of this stuff. But you and I measuring humans doing lots of different things, we end up with something that has aspects of randomness and aspects of determinism. And sort of teasing those apart is, to me, the business of complex dynamic systems research, trying to figure those out. So let's look a little bit more about sort of variation in biology. We see that biological systems have statistical self-similar sort of fractal properties. And here these are spatial properties going from nerve cells in the eye that no matter the scale that you look at, you see the same kind of structure. And also for blood vessels or airways in the lungs. And this is a valuable property if you think about what's going on here. These are all involved in gas exchange and or sort of exchange of liquids too. Having this sort of properties means that you have a lot of surface area for a given volume. So that's spatial fractal properties. There's also temporal properties that are fractal. And here you can see currents through ion channels in beta cells from the pancreas, that if you zoom in with a sort of a higher temporal resolution, you see the same structure. You see this sort of fractal properties. So that's at the sort of a real low level at within us humans. Let's see what we see at a more behavioral level of humans. And there's a technique that we can use to calculate this and try and tease these fractal properties apart. And this is called detrended fluctuation analysis, DFA. So what DFA allows us to do is sort of quantify a fractal structure in a time series. And this is how it's done. You take your time series and you break it into windows. Here you sort of see going from 1 to 51 is our first window, and these are all the same sort of size windows. We detrend each window with a simple line, and then we compute the fluctuations around that detrend, around that line, so around the detrended data. So we're seeing essentially a root mean square, an error, a dispersion around that best fitting line. And we repeat this process by using different window sizes. So we do a wide range of window sizes, doing exactly the same property each time. When you do that, you then you plot on the x-axis the logarithm of the window size against the logarithm of the root mean square, the fluctuations. And you get then data which is going to sort of give you something that you would calculate a slope to, and that's called alpha. That's the property you're getting. If you get an alpha that is 0.5, so this is a slope of 45 degrees, and so this is basically saying there's this equivalent increase in root mean square fluctuations with window size, then you see that the data can be equivalent to uncorrelated white noise. If you get an alpha that's less than that, you have what's called statistical anti-persistence. So you can think of this as being a fast stride time followed by a short stride time followed by a fast. Or if you go above 0.5 alpha, you get what's called statistical persistence. So here you see sort of drifts. Longer stride times are followed by longer, by longer, by longer, and then you'll drift down to shorter by shorter. Okay. So the idea here is that fractal properties would be revealed by values that are above 0.5. If you get a slope up towards 1, it's sort of considered a pink noise spectrum that you're looking at. And so it's suggesting that there's a range of temporal scales that are present in your time series. So here's an example from a young subject and a healthy subject. So it seems like looks like there's some differences in those traces, but it's tough to distinguish those with simple measures like mean standard deviation. But if we do a detrended fluctuation analysis, what we see is that young adults produce an alpha that is greater, closer towards sort of a pink noise values. So suggesting that there's more fractal-like structure, there's greater temporal scales within their time series. So that sort of goes against our original concept of, we just assume that variation in our behavior is just normally distributed and white Gaussian noise. However, what's interesting is as you age, you tend to drift down towards a white noise process. You seem to lose some of the temporal scales that we're seeing there. This is also seen with individuals with disease. They tend to also have a shallower slope alpha. So detrended fluctuation analysis is helping us to sort of reveal some structure in the variability that we wouldn't otherwise be able to detect. Now let's look at another way of looking at structure in time series. So here we have a time series that we've processed using what's called sample or approximate entropy. They are essentially equivalent measures. The original was approximate entropy, and that has been updated by other researchers. They've made some tweaks and to try and fix a couple of problems with the original approach, and called it sample entropy. And what it does is quantify predictability or regularity. So can you predict? And the idea is if you look at, and here we've got an example of a data point at, so this is heartbeat number 44, and you look at how does it change between 44 and 45. And then you just raise the question is, how often does that happen? If it happened a lot, then you would have a nice predictable signal. So it's relative to how much data you've got. So you're able to compute a probability. How many of these sequences do you see repeated? Of course, I've just shown you we have variability. We never get the same values twice. And so you need to have little windows around your values. And so that's what you're seeing here. The solid lines around value for the 44th heartbeat and the dashed lines around the 45th. And so what you're looking for is repetitions of these sequence of data points throughout the time series. And I've sort of indicated those in C. You count them up, count up how many possible sequences there could be, and you repeat this over and over again for every sort of pair of data points. And then you repeat it again. You can do a sequence of 3, and you can do a sequence of 4, and you can carry on if you like. And so there's also techniques that track that down and look at how things changes with sequence length. So this is another way of trying to say, you know, is there structure in this variability? Is it predictable? Is it regular? Or is it highly unpredictable? And when you calculate the sample entropy scores, to give you some examples here, if you have something like a sine wave, you get that's obviously a highly predictable signal. The vectors are repeated, the sequence of data is repeated over and over again. You're going to end up with a sample entropy value close to 0. If you have white Gaussian noise, where you can't predict anywhere through the time series, you're going to have a sample entropy up around 2. And if you are looking at something in between, you get, you know, sort of like the human isometric force data, you see some variation there. There is some predictability in that sort of signal. So the sample entropy helps us look at this predictability, regularity, and we can apply this to many different time series. Here is an example I've got from the data I showed you before of heart rate data from a healthy individual, an individual with congestive heart failure. When we looked at these time series, we thought they just didn't look right. They look different. And mean and variance are not different, but we can see sample entropy readily distinguishes these two. The congestive heart failure is a lot more predictable, regular signal. And that's what we've essentially, the field has discovered, that if your heart rate is highly predictable, that is a bad thing. The general understanding, I guess the theory of what that might be saying, is suggesting that we are losing timescales within our the variability of our heart rate, and means we are less adaptable. We're less able to change with the environment, things that are happening. And so this sort of congestive heart failure, we've got this rigid structure in here is sort of problematic. So that's a sort of second major approach to looking at interval ratio data and trying to tease out sequences or patterns in the data. So I'm going to now turn to a study that I did with Dr. Joanna Garner. And we were asked the question, to what degree do structured dynamics lie beneath visual gaze sequences? And do these vary with learning strategies? So here you'll see this sort of an application to nominal data. So we have participants study the heart. It kind of fits in with some of the previous data here. That was all accidental. But we've got these different images that they saw. So different sheets. They looked at A, talking about blood flow through the heart. You've got B, the structure. B is the blood flow, and C is oxygenation. Right, description. They were given a piece of paper and a pencil that they could take notes on. They were not told to take notes. They were just given that paper and a pencil. It was there. They were given instructions. There was a timer, and they were told to study, and they were going to do a test on this information. And they had up to 10 minutes, but they could decide when to quit. So we were wondering, you know, where do people look, and is there structure in those gazes? So what Dr. Garner did, because I'm not claiming this bit because this was a lot of hard work, was go through the videos and identify at each second where was the participant looking, and simply coded it with a letter. Code it with anything you like, but we got a code of a letter, and that's done. It's exhaustive. It is just essentially what is the most obvious item they're looking at that period of time, or and so that's coded like that. Okay. And you can look at some fairly sort of, you know, obvious, I think, measures. We can sort of say, what is the total task time? People take the full time or less. How long did they look at an object before transitioning? And what was the proportion of time that they spent looking at the different things? So the sheets, instructions, elsewhere, and then sort of counting up the number of transitions that they made. Right? So if you think about what could be happening here, people could be, they could be looking at sheet A all the way through. They could then switch to sheet B and then sheet C and be done. Okay. Or there could be some sequencing here, but you know, these people could be nice and deterministic, and they might go A, B, you know, all the way through, and then B, C, all the way through, something like that. They could be nice and simple. So we wanted to see how can we detect any structure in there. And I am all for plotting data, as I think you should have already seen from my slides. We used symbol sequence plots to get a sense of what people were doing. So in a symbol sequence plot, what we've done here is we're plotting essentially a black bar at each second for the different materials that they looked at. So you can see an individual here begins looking at E. If I remember that right, I think that's the instructions. Then they go to A, and you notice they never come back to the instructions. They go to A, look at A for a while, switch to B, and then you start seeing some more fluctuations as they go through. So this is a sort of, you know, so we can see, I think these simple sequence plots are a useful way of trying to start to see some structure that might be emerging there. We ran this through what's called orbital decomposition, and there is readily available software online. Dr. Garner and I have written a chapter about how to do this analysis, so the reference will be later if you want to get into this. But essentially, it requires the sequencing, so you have to provide the sequences. And what we did was push those into this sort of software. Now, we decided we could have included every single second, but we decided to go with independent of duration. So we were looking at sequence changes. So we put in for this data EA, BA, 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 and then so on. So that's what's going into the analysis. And orbital decomposition then is trying to detect structure in this sequence of nominal data. One of the variables it computes is Shannon entropy. And we can see that on the left-hand side for the plot that I've already shown you. This is a low Shannon entropy score. In fact, the lowest of anyone in the whole study. So fairly simplistic sort of pattern that this person is doing. And on the right-hand side, you see somebody that is doing something very different. And this person's doing, this person is achieving a sort of a high Shannon entropy score here with the sequence. And one of the things to particularly note is that this person also took notes. Okay. We can start to see differences between those. Right? So that orbital decomposition looks at recurring sequences and repeating sequences. Right? So ones that immediately recur and just general repeating sequences, and it teases out you know, is there a pattern there? How consistent are those? What we found was that the entropy scores were higher for note takers than readers. As I said, there was no instructions that they had to take notes. This was a choice that they made. But those that, when they did decide to take notes, they had greater novelty in their visual gaze. Now, there wasn't any difference in performance at the end on the test, so I can't say that having greater novelty in your visual gaze makes you a better learner. There's other things that could be going on in there, and of course differences at the initial knowledge going into the study. But we overall see the gaze complexity varied according to the task strategy. And orbital decomposition gives you Shannon entropy, also trace matrix topological entropy. And you can see that both Shannon and topological entropy are both greater in the group that were the note takers, also leading to a less stable attractor sort of space that you're seeing, which is quantified by a Lyapunov dimension. So the note takers seems to be associated with a more complex gaze sequences. Those are more sort of complex and immediately repeating gaze sequences. And so I think what we're posing here is that we starting to look at the dynamics of metacognition. So here, taking this sort of complex dynamic systems perspective, we're really saying, what is the dynamics? What is the sequence that's underlying a human behavior, in this case, studying? So to summarize sort of that study, we saw the gaze sequences and organization differed according to a global task strategy in self-regulated learning. And that's the sort of reference for that chapter where we put that together. So just to sort of summarize then, hopefully what I've persuaded you is that the time series analyses reveal structure in variability. And I've given you some examples there. There's ways you can look at the plots. You can do simple time series plots. Look at the frequency distribution can be informative. Recurrence plots can tell you about structure and sequencing, and symbol sequence plots also reveal that too. Time series analyses that I sort of introduced here, either for interval ratio data, we've gone through detrended fluctuation analysis that is able to sort of quantify, since you look at structure, we can distinguish whether we're producing uncorrelated white noise or something that is more sort of pink noise-like structures or something in between. And also sample entropy, which is a way of essentially raising the question, is our time series sequence predictable? Can we predict the future from the current sort of values? So that's another sort of useful metric. And then when you're looking at nominal data, we've got this orbital decomposition, which gives us measures of Shannon entropy and also topological entropy that can reveal aspects of the complexity in those time series. I'm also hoping that you sort of see that there's benefits of looking at individuals here, that it can reveal individual dynamic processes that are occurring and how those are different across people, but also how you can tease apart different groups using these kind of techniques to using the sort of standard statistical difference approach. I'd like to thank you very much for your attention, and I'm very happy to answer any questions you may have.
Thank you so much, Danny. Thank you much, Dr. Russell. I'm going to put in the chat the Jamboard once again. So for those of you who are just joining us or join us throughout, feel free to go there to post your questions. I'm actually going to share my screen, and Dr. Russell, we can talk through potentially some of the questions that are here, if there's anything that that speaks to you potentially, or I'm happy to choose one if you like.
Oh, no, I'll go for them. All right. Variability is always adaptive. Ooh. No. So I think I think there are elements where variability is not necessarily adaptive. I mean, we see that there can be large amounts of variability. This can cause you problems. So if you've got a sort of a noisy output, I do think, though, the other thing that I'm trying to highlight here that maybe wasn't clear is, so when we look at variability, I would say that standard deviation or coefficient of variation are measures of the magnitude of variability. These other measures that I've been talking through here, DFA, sample entropy, are looking at the structure of variability. And so one of the sort of main ideas is that a more sort of maybe more sort of certain characteristics of variability are adaptive. We are testing that directly. I mean, I would say I'm one of the skeptics in my area, because I think people have just assumed this is true. And students of mine have been studying this, and we'd be finding a less clear cut this sort of general idea of variability being adaptive. So I know it's a sort of a long answer, but I think I think we need to be careful about that as a sort of a general premise. But I'm definitely sympathetic to there are aspects to that. Okay, what are some usual plotting techniques? Okay. Okay. Yeah, so I mean, I think the ones I've highlighted there are useful. To me, time, I like to begin with the time series. So just looking at data over time is helpful. I think you could see some of those, that there was already structure, like in that congestive heart failure patient. So there's that you can do. Recurrence plots can help to sort of see whether you've seen changes you can see whether the sequencing how that sort of is related, are they correlated? Whether that changes over time, I think would be then interesting. You just sort of have to do a sort of a dynamic way, which you can do a recursive plots and look at how they change over time, look at the dynamics of those. But hopefully at least some of the plots I've shown today give some answers there. Yes, I did not provide any references. I will do that. So I will send a sort of a copy of the PowerPoint, and we'll include references there so you can check those out. Okay, so I'll just read it out so everyone knows where I'm at. I'm seeing how this type of analysis works well with biological or chemical processes, by having a hard time extrapolating this to analysis. Social complex systems involve human interactions. So I think, human interactions, I mean, there's obviously the gaze data that we looked there. You could look at gaze sequencing. It was, if you looked at parent-child, interesting to look at the gaze sequences there and that structure, and you'd be able to tease out how the sort of dynamics of that. Of course, if you're in a classroom situation, it could be between a teacher and a student. Any way of putting a code, to me, if you can come up with categories, which was why I sort of put up the example at the end of the gaze data. You can apply orbital decomposition to any kind of categorical data. So if there's any way that you can categorize the behaviors, then I think that opens up all of that orbital decomposition. Yeah, you're analyzing individuals' data. How do you make inferences about groups of individuals with similar characteristics? Great question. Okay. It is, to me, a problem in our literature. So sort of, first of all, say that, and I say our literature, sort of all of us looking at humans, we tend to compute groups and then we average across things. So, for example, there is a power law of learning that has sort of stood the test of time for quite a while. But when you actually start looking at individuals instead of group average data, you see that you don't see power law of learning. That's a group thing. So you've actually, power law has got nothing to do with how individuals learn. I'm being a bit strong there, but I think it's very, we need to see what's going on in the individual. And when you average across individuals, you looking at the gaze data there at the end, I showed examples of 4 different participants looking very different gaze patterns that, you start averaging across those, you see very different. It would just, obliterate the real data that's actually there. Having said that, throughout all of the work that I've shown here, what we have done is take metrics from individuals, and then we've gone and run standard statistics to see, are there statistical differences in DFA between groups? Are there statistical differences in Shannon entropy between readers and note takers? So there are ways of sort of doing that, but yeah, I highly recommend looking at the individual data. Very important point. All right. How transferable are the findings from the treadmill to movement in other terrains like hiking? Okay. So, I mean, some of the argument is that this, variability, and this is where the adaptive idea comes in, is that when you're hiking, you are not producing the same steps every time. The belt is not at a constant speed. And so therefore you need to be adaptable and changing these steps. So some of the idea is that if you think about that, we've evolved to be able to have these variable steps that meet the requirements of the environment, then we have an adaptive system that produces that variability in our gait, that we can't get rid of it even when we're on a treadmill that's producing this nice, consistent, smooth speed. One or two standard phase plots, EEG learns. Did you examine space plots using your data? Yes, and not particularly this data, the ones I showed you, but yes, I do phase plots too. Yeah, to me it's useful. So we also, so the graph I showed early of the knee angle during walking, we have a time series of motions of different joints of the center of mass of the body, and we use those to create, reconstruct embedding state space, and from that then we get local dynamic stability measures. We get correlation dimension, which we've done in gait, and punchline for some of that research is when you're walking at your preferred speed, your knees are most dynamically stable, and the correlation dimension is lowest. So your leg motion is essentially at a preferred speed is the simplest it could be and the most stable. Not true for the rest of the body though. These things are complicated. Have I hit Joey? Is there any others I should?
Looks like we got one more at the top.
Okay.
I don't know.
So what's your plotting and analyzing behaviors do you, and how you do your model the elements that oh, okay. Yeah, so I think we're just beginning to really explore that. So as I said, with the looking at data from walking, there's been two sides in the field. Some people are focused on the stride times or stride lengths, so they just get a value for each stride, which would be a right heel strike to a right heel strike, and then they do DFA sample entropy on that. The other side in the literature gets trajectories of joints over time, so like 100 times a second, usually over potentially minutes. And we look at those through local dynamic stability, correlation dimension. What we've done very little of is condensing that and seeing relationships between those. I mean, that to me is absolutely where we need to go as a field. I would say that my students and I are sort of one of the few people who are kind of doing both, which I think is people have seemed to have adopted one or the other approach. So I think my answer is let's hold on and see if we can try and figure that out.
Do we have any other questions or comments that people want to make while we're here? Still have some more time left. Okay. Well then, first, thank you. Something that I've come to really appreciate, and I know that my advisor, Dr. Api Kaplan, who's here on the call with us this evening, and I talk about a lot, is the value of disaggregating data, looking at the individual over time, and how much more you can see, and how much of that variability really does come into effect. So thank you for stamping that idea this evening.
Very welcome.
Sorry, let me get myself together. So before
While you do that, Joey, I
Yes.
just want to say thank you as well, and thank you everybody for coming. And we actually have 3 more of these webinars scheduled before the annual meeting in April in Chicago. So for those of you who are coming to Chicago, we cannot wait to see you in person. And for those of you, whether you're coming or not to Chicago, please join us for the other 3 webinars. And I think, Joey, you have the details of those.
I do, yeah. So first, can we all please join me in thanking Dr. Daniel Russell for his presentation this evening? Snaps, claps, all the things. So thank you all so very much for being here, Dr. Russell. Thank you so much for your presentation. I am very excited to share that our next presentation is going to be the next session is Wednesday, February 22nd in 2 weeks at 12 p.m. Eastern time, where Dr. Helene Pennings will present on state space grids. And you can register by clicking on the link that I just posted in the chat. If there's no other questions, I want to thank you all once again for coming. Thank you, Dr. Russell, for providing such an informative and enriching conversation and presentation this evening. And I hope to see you all in 2 weeks at 12 p.m., February 22nd.
Thanks, everybody. And thank you, Joey, for being the